Thank you. Our next speaker is Dr. Eduardo Marbán from Cedars-Sinai.
Good afternoon. I'm going to talk today about products of cells that can be used for cell-free therapeutics. And the disclosure here is relevant because I'm a founder of this company, Capricorn. I'm going to talk about some of its technology at the end and how the company has been very helpful for advancing the overall research program. So this is a summary of 12 years of work on a cell type that we first isolated in my laboratory at Johns Hopkins, where I was until 9 years ago. And we gave it a process-derived name, cardiosphere-derived cells. The idea was that the heart contains its own reservoir of progenitor cells, and these might be useful therapeutically. And we went about a strategy of primary culture, whereby we took small bits of a cardiac tissue biopsy and put them into dishes that were coated with fibronectin so that they would adhere, and under these conditions, cells spontaneously come out. We called these explant-derived cells, and those explant-derived cells could be harvested and replated on non-adherent plastic polylysine-coated dishes where they self-assembled into organoids called cardiospheres. And these three-dimensional microtissue organoids could then be replated to yield the therapeutic candidate cardiosphere-derived cells after 2, 3, or 4 passages. We described this in 2007, and since then the methods and the bioactivity have been reproduced by at least 26 independent labs worldwide. About once a month, a new lab somewhere in the world comes up with some confirmation of this technology. The cell type turns out to fulfill the criteria for a cardiac progenitor cell in the sense that it's multipotent and clonogenic, but that doesn't seem to have much to do with the mechanism of action per se, because the cells can die off and there are long-lasting effects, as I'll show you in a minute. So we tend to think of them more as cardiac stromal cells with extremely interesting regenerative properties. In terms of characteristics, they're uniformly positive for the TGF-beta receptor accessory subunit endoglin, CD105. They're negative for CD45 and all other hematogenious markers. They secrete SDF1, and they induce the secondary secretion of SDF1 via exosomes that contain a distinctive panel of microRNAs and other non-coding RNAs. They've been in the clinic now several times. The first clinical trial was Caduceus, sponsored by the NHLBI. This is a completed study. It was an autologous phase 1 study of 25 patients, randomized. 17 received autologous CDCs, and 8 were standard of care. Everyone underwent very careful phenotyping, and we got evidence of regeneration in the sense of increased living heart muscle in the patients who were treated with CDCs after a myocardial infarction, and this was published in The Lancet in 2012. This has now gone on to the allogeneic product, which we realized during the conduct of Caduceus was viable, and even in non-immunosuppressed patients. All-Star Phase 1 was a 14 patient first-in-human study. It's now complete. And the Phase 2 study involving 134 patients, which is a multi-center, randomized, placebo-controlled, double-blinded study, just completed enrollment. And these are using allogeneic CDCs, post-MI in patients with mild heart failure. We've also recently completed a study called Dynamic, which is an open-label 14-patient study in patients whose only option would be transplantation other than this. And this is a positive study, which we will be describing in more detail at the TCT meetings. And finally, Hope Duchenne, treating the cardiomyopathy associated with Duchenne muscular dystrophy. And you've heard about Hope Duchenne this morning from Pat Furlong. Enrollment is complete in both this and All-Star, and so readout of the primary one-year endpoints should be next year. The mechanism of action, even though they are cardiac progenitor cells, turns out to be paracrine. And Victor Zhao, who's just spoken, was the father or the pioneer of the paracrine hypothesis for stem cell action. These cells turn out to be no different. They promote cardiomyogenesis, prevent cardiac myocyte apoptosis. They're anti-inflammatory and anti-fibrotic, and they also appear to have a variety of other beneficial effects, which I'll review in a second. But basically, the canonical stem cell mechanism, whereby transplanted cells would proliferate, differentiate, and cause new healthy tissue of donor origin, appears not to be the case, and instead the CDCs are transplanted and survive probably for 3 to 4 weeks, during which time they secrete factors that lead to new healthy tissue that's durable and present, but it's of host origin. So that begs the question then, is there a single entity that can mimic all of the salient benefits that we might be able to use for cell-free therapeutics? And we have begun looking at exosomes. These are bioactive nanoparticles that are secreted by all eukaryotic cells and are products of the endolysosomal pathway. They're 30 to 150 nanometers in diameter, and just to put that in context, an HIV-1 virus is 100 nanometers in diameter, and a ribosome is about 300 nanometers in diameter. So these are smaller than the smallest organelle. They're present in all body fluids, and by present, I mean tens of billions of exosomes per milliliter of a typical body fluid. And they're loaded with microRNAs and other bioactive contents. The payload is extremely specific not only to the cell type, but also to the conditions under which the cell is cultivated. And the reasons that we are excited is that, I'll show you evidence here, that CDC-derived exosomes mimic the effects of CDCs in the absence of cells, and blocking the production of exosome biosynthesis from the CDCs abrogates their benefit. So this is a myocardial infarction model in a mouse, and this is the natural history in light blue of what happens to ejection fraction, which is a simple measure of cardiac function over time after that model. If you put in exosomes from a fibroblast line from humans, they have no effect, but if you put in exosomes that were isolated from media conditioned by CDCs, they have a very positive effect. That positive effect is reproduced by CDCs themselves, but abrogated by incubating the CDCs in GW4869, an inhibitor of ceramide synthesis, which is required for exosome biosynthesis. It turns out, after many experiments, that we now have reassured ourselves that CDCs produce these effects, all of which are mirrored and recapitulated by CDC exosomes if delivered correctly into the tissue. Regeneration, antifibrotic effects, anti-apoptotic effects on cardiomyocytes, angiogenic effects, anti-inflammatory, and importantly, immunomodulatory effects. As I said, they contain microRNAs, and they not only contain microRNAs, but they contain a very distinctive profile. For example, in comparing by microarray the amount of microRNA 146A in C2C-derived exosomes versus exosomes from normal human dermal fibroblasts, there's a 260-fold excess in the CDC-derived exosomes of 146A relative to NHDFs, and others you can see are less plentiful. We chased 146A as a possible candidate of the bioactivity in the CDCs and their exosomes, and indeed, many of the effects of the CDC exosomes and the CDCs, the regenerative effects in particular, were largely mimicked by 146A, but selectively. It turns out that not only are there many microRNAs, but microRNAs are themselves, microRNAs being this part of the pie, only about 7% of the small RNAs within exosomes, and you can see there's a bewildering array of other things, including yRNAs and links and piRNAs. Interestingly, the NHDF exosomes contain a lot more of the starch, the tRNAs, than the bioactive CDC exosomes do. So we're busy figuring out what a lot of these other non-coding RNAs do, and we have evidence for bioactivity of the yRNA fraction, for example. So the working hypothesis is that no single RNA species can account for all of the benefits of exosomes, and that individual mirrors or other RNA species may prevail in any given setting. It's the totality of the contents that are required for full manifestation of activity, but in the totality, they hit many, many, many targets, so they reproduce the benefits of cells. It's not a small molecule-like effect. So salamanders were mentioned a second ago. We were intrigued by the fact that regeneration in newts is much greater than that in mammals. They can regenerate the entire retina or a limb or the lens, and they can even regenerate a transected spinal cord. So we wondered whether the newt biology might be somehow translatable to mammalian therapeutics. It's notable, though, that we separated from the amphibian lineage about 300 to 400 million years ago. So if there was some convergence, it would be somewhat remarkable. But we tested the idea that newt cells can make exosomes, and we asked, are the exosomes biologically active in humans? If you look at PubMed and put in newt exosome, there's 0 hits. So we were the first to look for these, and we find extraordinary bioactivity. And we used as a source for the exosome a naturally immortal cell line called A1 that was derived from the newt blastema, which is the healing stem cell-laden cap after amputation of a limb. It turns out that newt A1 cells make beautiful exosomes. They're chock full of RNA. They contain 8 times more RNA per particle than mammalian exosomes. The distribution of non-coding RNAs is, again, bewildering, but somewhat reminiscent of that in CDC exosomes. They markedly stimulate rat cardiomyocyte proliferation in vitro, and in vivo, they have regenerative benefits. They improve viable area, decrease scar area, and improve function after a myocardial infarction. So will therapeutic exosomes ever reach the clinic? Here, Capricor comes into prominent play. Academic competence is limited, and at some point you have to transfer the technology collaboratively in the best scenario to a company that can take it to the next step. I co-founded Capricor in 2005 as then a spinoff of Johns Hopkins. It became listed on NASDAQ last year. It now has approximately 40 employees, but the distinctive features that I think may be important lessons for thinking about what we might do better in regenerative medicine is the company never took any VC money. 100% of the management is either MDs, PhDs, or PhDs, including the CFO, who's an MD, PhD. It's received very extensive peer-reviewed grant support from CERM, NIH, and DoD. And the scope is soup to nuts. It goes all the way from discovery through development, manufacturing, regulatory and clinical trials design and management. And Capricor has reduced to practice in a GMP-compliant fashion not only the processes for making clinical grade CDCs, CAP 1002, but for taking media conditioned by CDCs and making CAP 2003, which is the clinical exosome product, which they hope to have in the clinic sometime next year for fibrotic and inflammatory diseases of the eye. So in summary, cardiosphere-derived cells are viable therapeutic candidates themselves. I don't want to throw the baby out with the bathwater, but they have all of the limitations of cell therapy, fragile living entities, hard to manufacture, et cetera. Exosomes mediate and reproduce the benefits of CDCs. Interestingly, newt exosomes are bioactive in mammalian injury, and we're pursuing this as its own category of therapeutic agents. And CDC-derived exosomes are in clinical development for diseases of prominent fibrosis and inflammation. And these are the important people that did the work. And I just want to highlight that purple is Capricor and white is me, but we've enjoyed funding from NIH, California Institute for Regenerative Medicine, and the Department of Defense to move this forward. Thank you very much.